I'm Jarrod, co-founder of IonOpticks and head of product development. Day to day, my role is taking ideas from the research laboratory and guiding them through a product development process to release into the market for the benefit of researchers worldwide. The most interesting thing about my job is seeing the impact that our products can have in the research community, talking with customers, hearing about how our columns have assisted their research and the outcomes and the discoveries they've been able to make. What I love about proteomics and the field of mass spectrometry is the potential for discovery. We're really only at the beginning of an enormous book, the first chapter of what we can discover in medical research and the impact that can have on human health. The reason why IonOpticks is so important in the LC-MS field is that for many years the focus was on developing mass spectrometers as the solution to getting deeper into samples and getting more identifications. What was neglected for a long time was the chromatography, the separation of samples. Now with the developments that IonOpticks has made and the better definition you can get and better separation you can get out of samples, what we've discovered is that the existing mass spectrometry technology can actually be utilised far more efficiently. We can see gains of 20, 30, 40, even 50% in terms of identification just by attaching better separation to that existing mass spectrometer. And I think that's really powerful because it opens up a world of discovery for the equipment that already exists in people's laboratories today. Getting deeper into samples can mean the difference between finding a biomarker and missing a biomarker. It can be finding a drug target and missing a drug target. We're not talking about just doing part of a research. We're talking about black and white. It can be the difference between a successful project, a successful drug development, a successful diagnostic for a sick patient, and not discovering that. And when researchers are spending years of their life focusing on these questions, they need the greatest support and the greatest definition in their samples to achieve that. I think the thing holding proteomics back from becoming more mainstream is the ease of use. It still is a little bit piecemeal. It still is a little bit put together. There's no universal solutions from different vendors. There's no one option that a life science researcher can turn to if they want to do proteomics work. They still need to engage a technical person, someone with years, sometimes decades of experience to help them achieve that. I think the focus of the field has to be on making these things more accessible, making these things easier to use, making it simpler to go from your sample analysis and then a result at the end of your experiment. And until we achieve that, I think we're still going to have a separation between the field of proteomics and general life science. My hope for the field of proteomics over the next 10 years is that it goes from the bench, from the academic lab, from the research institute, more into the clinic, into diagnostics, into mainstream use, where those discoveries that are being made at the moment in an academic institution on patient cohorts from biobanks and samples have actually made it into meaningful diagnostics, where people actually rely on proteomics to tell them if they're sick, to tell them when they're going to be sick. I think that would be a great advancement. And I think IonOpticks can help with that by allowing the analysis of those samples, both now and in the future, to be done with much greater definition and much deeper and more robust, so you can have confidence in the results that you're seeing. I see my role in the community as an enabler to take ideas that can have a real impact in the research lab and to a degree democratize these products so they can be distributed more broadly. The idea of IonOpticks originally was that we took a technology that was only available to a very small number of labs and used in very discrete settings, and something that was difficult to use, cost a lot of money, cost a lot of time, wasn't particularly reliable. And making that available to the broader community, I think personally that gives me a lot of satisfaction, and we can already see the impact that is happening in the research community. As a dad of a new daughter, I want to create a world in the future where she has more confidence when a doctor is telling her the outcome of a result, where she has confidence that when she's been prescribed X, Y, or Z, that that's actually targeted to her illness or disease or condition, not something that's prescribed more broadly. That sort of precision approach, I think, is what we can really enable going forward, and what will help her and everyone else in the community to live happier and healthier lives.